Namaskar, welcome to our channel by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, today's topic is linkage and recombination as well as linkage frequency and recombination frequency. We have learned different laws of Mendel's, also exception to the Mendel's laws. Now, today we shall learn about linkage frequency as well as crossing over and crossover frequency. Linkage and recombination as well as linkage and recombination frequencies. Thomas Hunt Morgan studied this linkage phenomenon. Thomas Hunt Morgan studied this linkage frequency on Drosophila melanogaster. Drosophila melanogaster is a fruit fly. Why he selected for this fruit fly for his experiment is very important to know as a part of this experiment to begin. So, one that he found short period of maturity so that result can be obtained in short time. This is the first reason. Second reason, that male and female can easily identify, and that was very important. Third, that leaving more progeny or producing more progeny so that it helped to observe that character in most of the offsprings obtained from it. And last one, that easily observable traits or characters under simple microscope. You will no need to have the compound microscope. Therefore, he selected, or his team selected, Drosophila melanogaster for their experiment. I hope this you got it. Question can be asked for 2 marks in board. Why Drosophila melanogaster was selected as a example or animal for its experiment, or why Thomas Hunt Morgan has selected Drosophila melanogaster for his experiment. So these are the 4 points that you have to mention in as an answer in examination. Now, they have observed, Thomas Hunt Morgan and his team members decided to carry out an experiment on Drosophila melanogaster, and therefore they have selected as an example for their experiment that traits they have selected to be observed was trait that selected two traits. One, that color of the body and color of the eye. Color of body, and second was color of eye. These were the two. Hence, this was a dihybrid cross. So let us study how they have carried out that experiment and what observation they did made. So here we have that another page we can use for studying that experiment. See, brown body and red eye, fruit fly cross with yellow body and white eye. These were the parents for their experiment. Now, what are the genotypes? Genotypes are capital B, capital R being dominant over that yellow body, small b, small r, small r. When they cross it, that with the gamete capital B, capital R and small b, small r, in F1 generation after fertilization, they obtain only brown body and red eyed, brown body and red eyed houseflies, all 100% in F1 generation. As they were knowing that in the hybrid cross, in F2 generation, according to the Mendel's, that phenotypic ratio which was expected, that is expected ratio, phenotypic expected ratio, was 9:3:1. What was the 9? 9 red brown body and red eye was expected. Here yellow body and white eye. That one. What was about recombination is that brown body and white eye as a recombination, and another recombinant yellow body and red eye. These are the recombination, as they were not parent. Brown body, red eye was parent, but brown body with white eye and yellow body with red eye as a recombination. It was expected, but what they did observe, and they got ratio, that they got 7:1:7. See the number of members, which supposed to be parental, found more than that of recombinant. So this was the percent. This was the percent, that 48.7, and recombinant was just 1.3 and 1.3. So altogether, that if we consider this recombinant was 2.6%. Here we can see that percentage which obtained for parental combination is 48.7. So altogether, total it can be, as if we take it percentage, it can be 97.4. Yes, it is 97.4. So parental combination obtained was very high than that of the recombination. Since it shows that only 1.3% that is the recombination. That means that genes which are present away from each other, they are just 1.3 mach unit distance away. So as to understand, if we draw these two chromosomes as a homologous member of chromosome, and these genes are present on that locus are very close, that is 1.3 mach unit distance away. So here, as that brown body, if we say capital B, so yellow body will be small b, and red eye is capital R, then white eye will be small. So this distance is just 1.3 centimorgon. This is just 1.3 centimorgon. It shows that these genes are very close to each other, and hence they inherit as it is in their parental form, what we call that linkage. Therefore, they conclude by saying definition of linkage that when two or more than two genes present very close to each other on homologous pair of chromosome on different loci, and inherit as it is in their parental form are known as linked genes, and the phenomenon is known as linkage. What exactly they compare? That according to Mendel, that phenotypic ratio has to be 9:3:3:1, but what they obtain 7:1:1:7. That means that 7 for brown bodied and red eyed and 7 for yellow body and white eyed. This shows that more parental combination than that of that new combination with brown body and white eyed or yellow body and red eyed. So finally they conclude that because of this ratio, they conclude that these genes, that genes for brown body and red eye as well as yellow body and white eye must be very close to each other. Therefore, they could not cross over and obtain recombination, a new recombination, and number of recombination which were observed, that was very less in number. That means that genes are linked genes and inherit as it is in their parental form. Therefore, that genes they called as linked genes, and phenomenon they called as a linkage. I hope you understood well. Even if you are in doubt, you can write in comment section. I appeal you to subscribe our channel and like our videos, as well as share with your friends as many as you can. Press the bell icon to receive notification as and when new videos are uploaded. Thank you.